Welcome to the video presentation of our short paper, Detection and Visual Analysis of Pathological Abnormalities in Diffusion Tensor Imaging with an Anomaly Lens. This is a joint work between the University of Bonn and the University Children's Hospital in Tübingen, Germany. The diagnosis of disease often involves taking measurements and comparing them to a normal range. For example, many modern digital thermometers visualize their measurements of body temperature with a color code that indicates whether they are within or above the normal range. The goal of our work is to achieve something similar, but in the much more complicated scenario of diffusion tensor imaging, or DTI. DTI is an advanced medical imaging modality that is mostly applied to the brain. Unlike a thermometer, it requires not just one global value, but spatially resolved images. Therefore, whether or not a measurement is within normal range depends on its location within the brain. Moreover, it captures not just a single value per location, but rather a 3x3 symmetric matrix, the diffusion tensor, which corresponds to an ellipsoid at each voxel. This quantifies the directionally dependent diffusivity, and it is sensitive to pathological changes that affect tissue microstructure, such as damage to the fiber tracts that connect different brain regions, or the brain and the rest of the body. A comprehensive visual analysis of this rich information is our challenge. In current clinical practice, such datasets are screened for pathological abnormalities by viewing two derived scalar volumes. Mean diffusivity, which is shown here, indicates the overall diffusivity in a voxel averaged over all directions. Fractional anisotropy is the second one and indicates how much the diffusivity depends on the direction. This current practice has two important drawbacks, which are addressed by our system. First, it relies on the viewer's implicit mental model of normal anatomy. In the example we see here, the human analyst needs to know that FA in healthy subjects is typically higher in specific regions to realize that it is unusually low in the patient that is shown here. Our system establishes an explicit computational model of what constitutes a normal DTI scan, and this makes it possible to highlight anomalies and guide the user towards them. Second, fractional anisotropy and mean diffusivity only correspond to 2 degrees of freedom in the diffusion tensor, while our system permits a more detailed analysis that accounts for all 6. Therefore, even though our system only requires going through the data once instead of twice, it still conveys a more comprehensive picture of the full tensor information. I will now illustrate our proposed novel workflow with an example. Our model of normal brain anatomy is constructed from a group of healthy control subjects, which are brought into spatial alignment with each other and with the patient that we would like to investigate. Deviations from that control group are quantified with an anomaly score that is visualized to guide the viewer towards slices in which abnormalities have been detected and to highlight their locations within those slices. To find out why a specific region has been highlighted as anomalous, it can be selected by placing an anomaly lens with an arbitrary position and radius. Colored segments around the lens indicate how different aspects of the diffusion tensor contributed to the anomaly score within the lens area. For their interpretation, consider the ways in which diffusion tensors can differ. As symmetric 3x3 matrices, they have 6 degrees of freedom, which correspond to norm or overall diffusivity, fractional anisotropy or FA, indicating the overall extent to which diffusivity is directionally dependent, mode or type of anisotropy, indicating whether diffusion happens primarily along a line or a plane, and 3 rotational degrees of freedom, which we group together to ease interpretation. In our example, the fact that the green and the orange segments around the lens are the largest indicate that changes in norm and FA contributed most to the region of interest being detected as anomalous. Clicking on one of the segments color codes the respective deviations within the lens area with a diverging color map. In this example, clicking on the orange segment for FA colors the interior blue, which indicates that values are below the normal range in our control group. Clicking on the green segment for tensor norm colors the interior of the lens red, indicating that values are elevated. These findings are consistent with a loss of tissue integrity in this region or a demyelination of the neurons. Finally, our tool considers not just the variations in individual tensor aspects, but also covariations between them. All pairwise dependencies are visualized as edges in a covariance view on the left. As we just saw, increased norm went along with decreased anisotropy. The strong blue edge between norm and FA indicates that this is in agreement with the control group in the sense that even in healthy subjects, above average tensor norm tended to go along with below average anisotropy. Before we look at further examples, let us briefly consider the mathematical model that is behind this visualization. As an anomaly score, we use the Mahalanobis distance, which is a well-established measure of distance between a multidimensional point x and a distribution that is described by a mean vector mu and a covariance matrix sigma. For this, we embed the diffusion tensors isometrically into 6-dimensional Euclidean space. Means and covariances are computed separately in each voxel after bringing all volumes into spatial alignment with a specialized image registration algorithm for diffusion tensor images. Our visual design relies on a decomposition of the anomaly score. It is based on the invariant gradient and rotation tangent framework for diffusion tensor analysis that was developed by Gordon Kindelman and his co-workers. Given the local mean tensor, it constructs an orthonormal basis Q in which the coefficients of deviations from the mean become aligned with the gradients of norm, FA, tensor mode, and rotations around the 3 eigenvectors. Because Q is orthonormal, this change of basis does not change the magnitude of our original anomaly score. However, writing the Mahalanobis distance in that basis allows us to decompose the resulting double sum into diagonal terms that reflect deviations in norm, FA, mode, and rotations, which are mapped to the rim of our anomaly lens, and off-diagonal terms that reflect the agreement between partial correlations between those aspects of the tensor in the control group and the corresponding covariations in the patient. These are visualized in the covariance view. Further details, especially how we group together the rotational degrees of freedom, are given in our paper. Together with collaborators from the University Children's Hospital in Tübingen, we used our system to analyze diffusion tensor images of patients with metachromatic leukodystrophy, a disease in which an accumulation of sulfatides damages the myelin sheets around neural fibers in the central and peripheral nervous system. The increased overall diffusivity and decreased anisotropy that we observed in our introductory example is a typical sign of such demyelination and is in line with observations that others have reported in the literature. Our system identified similar changes in all MLD patients, but revealed individual differences in their localization and in the relative magnitude with which norm and FA contributed. While increased norm was the dominant factor in our previous example, decreased FA is the strongest driver of anomaly in the geno of the corpus callosum in the sagittal region of interest in another patient. The screenshot also illustrates the tooltips that permit the quantification of our observations. Finally, we show a result in a third patient. Here, the covariance view highlighted a surprising covariation between fractional anisotropy and tensor mode, as highlighted by a strong red edge. Inspecting FA and mode separately by clicking on the corresponding segments of the lens shows that diffusion tensors in this patient were slightly more linear than average in this region of interest, despite having lower fractional anisotropy. This finding might indicate a region in which crossing fibers are unequally affected by demyelination, and this would have been impossible to observe in a traditional inspection that just accounts for mean diffusivity and fractional anisotropy. In summary, we presented an interactive visualization system for the detection and investigation of pathological abnormalities in diffusion tensor imaging that accounts for the full tensor information and provides guidance that is not available in the traditional workflow. Our tool consists of an overview visualization, an anomaly lens that reveals which aspects of the diffusion tensor dominate the overall anomaly in a region of interest, and a covariance view that highlights unusual covariation. Mathematically, our tool is based on decomposing the Mahalanobis distance, a well-established anomaly score for multivariate data, into individual terms that relate to deviations in tensor norm, FA, mode, rotation, as well as covariations between them. In future work, we hope to extend this approach towards even more advanced variants of diffusion imaging. With this, we would like to thank the German Research Foundation for providing the funding for this work and you for watching our presentation.